Part 0. Laboratory Principles. Introduction. Practicals in the subject of science have a very major role. In fact, the subject cannot be understood well just by learning the theory. When a student does practicals, the concepts become very clear and it becomes easy to verify the facts. Practicals are performed in a laboratory, and every student should understand some rules of working in a laboratory. Laboratories are well equipped with necessary material and help in clarifying doubts by performing experiments. Equipments. Students should come well equipped to the laboratory. They should carry with them the following: practical notebook and laboratory manual, pencil, eraser, sharpener, and scale. Instruments such as forceps, scissors, dissecting needles, blade or razor, brush, and dropper. A cotton duster for cleaning the equipments. Any other special equipment as suggested by the teacher. Students should come prepared with the background of the experiment. The instructions given by the teacher should be listened to carefully. Students should work in the laboratory in a very disciplined manner. Working tables should be properly set and systematically they should proceed. One should be very careful while handling glassware. Before using it, be sure that it is clean, otherwise it should be washed. Physiological experiments. The experiment should be set according to the instructions given. Observations should be made very carefully and conclusions should be drawn. In case the results deviate from what is expected, students must discuss with their teacher. Laboratory records should be maintained very well. They should be very neat and tidy. They should have blank sheets as well as ruled sheets. The blank sheet should be towards the right-hand side and the diagram should be drawn on it. Comments for the same should be on the ruled page on the left-hand side. A margin should be made on the drawing sheet, horizontally as well as vertically. The diagram should be labeled with neat lines. They should not cross each other. Labeling should be done on the right-hand side of the sketch. The diagram should be proportionate and should not be shaded. It should not have broken lines. The lines should be made in a flow. Laboratory maintenance. The laboratory should be kept very neat and clean. Stain should not be dropped on the table or floor. If it drops, it must be wiped off. The working tables and floor should not be littered. Extra material must be thrown in the dustbin. Study of permanent slides. Slides which are fixed under the microscope should be studied carefully. They should not be shifted. If there is any doubt, the teacher must be consulted. Diagrams should be drawn from the slide and compared with the manual and labeled. Temporary mounts. Mounts should be done neatly as per the instructions given in each experiment. The material to be mounted should be kept in the center of the slide. Study of museum specimens and models. The specimens and models should be studied very carefully. They should be observed from all angles and sketches should be drawn. The sketch should be compared with the diagram given in the lab manual. It should be correctly labeled. Microscope. In biology, there are many objects which are very small and they cannot be seen with the naked eye. Our eye cannot see objects which are smaller than 0.1 millimeter. To observe such objects, we need microscopes, which are magnifying devices. Different types of magnifications can be obtained with the hand lens, dissecting microscope, and compound microscope. A brief description of each of these is given. Hand lens. A hand lens consists of a biconvex lens mounted on a handle. It is of different sizes and different magnifying powers, and it helps in magnifying objects. Its magnifying power may be 2x, 3x, 5x, or 10x. Dissecting microscope. This microscope is used for seeing objects in which a very high magnification is not required. Sometimes it is also used for dissecting small objects. The lens that is used is normally of 5x, 10x, or 20x. The parts of the microscope are: base. It supports the other parts. Stand. It is short and cylindrical. It supports the microscope. It has a mirror near the base. Vertical limb. It fits into the hollow tube and can be made to move up and down with the adjustment screw. Folded arm. It is attached to the vertical limb and at the other end a lens is fitted. The arm can be made to move up and down as well as sideways. Stage. It is a glass plate. It is used to keep the slide or object. Clips are provided to keep the slide in position. Mirror. A concave reflecting mirror is present below the stage. It is used for reflecting the light rays onto the stage. Working of the dissecting microscope. The slide is kept on the stage, held in position with the help of clips. The lens is brought over the slide. Then, the vertical limb is moved up and down to focus the object. The light is adjusted with the help of a mirror. Compound microscope. It is a microscope which has a set of two lenses. Both are biconvex. An enlarged and magnified image is obtained. It has the following parts. Mechanical parts. Base. It is horseshoe shaped and provides support to the microscope. Pillar. It extends vertically from the base. Arm. It is used for handling the instrument. Stage. It is a rectangular flat plate on which the slide is kept. It has a hole in the center for light rays to pass. It also has clips to hold the slide firmly. Diaphragm. It is present below the stage and regulates the amount of light entering into the microscope. Condenser. It is used for focusing the beam of light on the object. Body tube. It can be moved up and down with the help of screws. Nosepiece. It is a circular metallic structure. Objective lenses, which may be 2 or 3, are attached to it. Coarse adjustment screw. It is a big-sized screw which helps in focusing the object. The body tube moves up and down. Fine adjustment screw. It is a smaller screw which is used for fine and sharp focusing. Optical parts. Mirror. It is present below the stage. One of its surfaces is plane and the other is concave. It is used for reflecting light rays into the microscope. Objective lenses. These are attached to the nosepiece. Their magnification may be 10x low, 45x high and 100x oil immersion lens. Eyepiece. It is fitted at the top of the body tube through which the magnified image of the object is seen. It is of magnification 10x or 15x. Working of the compound microscope. First of all, adjust the mirror so that sufficient amount of light enters the microscope. See through the eyepiece. The eyepiece, mirror, objective lens and stage should be very clean. The slide is kept on the stage, fixed with clips, and then it is focused with the help of the adjustment screws. First we see it under low power and then under high power if required. Correct way to use the microscope. Keep the microscope clean. Always hold it in an upright position with both hands. First, focus under low power and then high power, whatever has to be viewed. The objective lens should not touch the slide. Always move the draw tube upwards rather than moving it downwards.